Welcome to SPARQed's second module of the Working with DNA lessons. This module is an introduction to the specific steps you'll get to do if you come to SPARQed to do one of the DNA workshops. Ideally, you should work on booklet 2A while you go through this presentation. In this presentation, the learning intention is to apply your knowledge of recombinant DNA technology from Module 1 to the process of cloning a gene called GFP. At the end of the presentation, you'll be able to recall the process of making recombinant DNA, describe the purpose of cloning the GFP gene, identify the fact that there are factors that regulate the expression of genes, and describe the purpose of the steps in making recombinant DNA. In one of the SPARQed workshops, you can work with a gene called GFP. GFP stands for green fluorescent protein. The GFP gene is very popular in labs around the world. It was originally obtained from a particular species of jellyfish. It has been modified for use in research. The protein itself fluoresces when exposed to UV light, which is what makes it so useful. So, why do researchers like to use the GFP gene? Some people work with the GFP gene when it is attached to another gene. This results in the fluorescent protein being attached to another protein. The image on the left is from SPARQed's cell biology workshop, where you get to work with human cells that have GFP in their nucleus. The GFP protein here has been combined with histone proteins. Remember that histone proteins are found in the nucleus of eukaryotic cells. Some researchers also work with GFP attached to specific proteins in mice. They can then look at tissue samples under the microscope to determine where in the mouse's body their protein is located. You can see further interesting images at SPARQed. This is a diagram of the PGLO plasmid. PGLO is a plasmid used as a vector for the GFP gene. You'll notice that the GFP gene, shown here in bright green, is not the only gene on the plasmid. We've also got a gene for ampicillin resistance. This gene is useful for the process of transforming bacteria with the plasmid. In addition, we also have a gene called arabinose C. Arabinose C helps to regulate the expression of the GFP protein. Basically, the protein will only be expressed when we give the bacteria a sugar called arabinose. The other markings here on the plasmid map are sites where restriction enzymes can cut the DNA. So these are the names of specific restriction enzymes. You can learn a bit more about restriction enzymes in Module 3. When you come to SPARQed to work with DNA, you will learn a number of laboratory techniques. You will receive instruction about safety in the lab and how to use equipment such as micropipettes and centrifuges. You will then complete Part A, Part B, and Part C listed here. Before your workshop, the SPARQed teachers will prepare some bacteria by inserting the PGLO plasmid into the bacteria and then letting the cells grow and replicate overnight. Recall that transformation is the name of the process where DNA is inserted into a new organism. After transformation, bacteria were grown up on agar plates, which are shown in this picture. Then, a single colony of bacteria from the agar plate, and the bacteria now contains PGLO Plasmid, was used to inoculate some nutrient-containing broth. The bacterial cells then replicated in the broth overnight, therefore amplifying the amount of DNA present. We grew up 2 tubes of E. coli bacteria containing PGLO. To both tubes, we added an antibiotic called ampicillin. This ensured that only the bacteria that successfully received the plasmid from transformation will be able to grow. All the other bacterial cells will be killed by the antibiotic. Recall that the other gene in the plasmid is called arabinose C. It is part of a collection of genes that normally help the bacteria transcribe some digestive enzyme genes. Here, the arabinose C gene means that we only switch on the GFP gene when we add a sugar called arabinose. In the picture on the left, observe the 2 tubes under white light. You can see that they're a bit cloudy. That's because they contain a lot of bacteria. They grew up overnight. Now, observe the 2 tubes under UV light. Which tube is expressing the GFP protein? To which tube did we add arabinose? Now it's time for you to try the questions on page 6 of the booklet. Well done on getting through that. Once again, we hope it was useful. If you have any questions, please don't hesitate to be in touch.